So that was the primary structure. Nucleic acids also have secondary, tertiary structure, just like the proteins did. So without going through all the history of this, which is very cool by itself, but we don't have time for that. One of the important observations was in any DNA, actually in any nucleic acid, the amounts of A and T bases were always equal. We always had equal amounts of A and T, and that's, like, wow, different species, didn't matter. And there were also equal amounts of G and C bases. So back in 1953, Watson and Crick proposed the now famous double helix structure, which was found to be correct. And this accounts for the equality of bases and also for some other structural data. So there's different ways of representing it. This is more of a space-filling model, which is all very nice, but a little hard to look at. This is the top view of the space-filling model of the double helix. When we look at it this way, it's a little easier to see what's going on. The DNA has actually two strands, and here this orange one is going 3' to 5' end, and the blue one is going 5' to 3'. And so they have an anti-parallel nature. It's a head-to-tail arrangement. And so this is a little bit like a spiral staircase, where you've got the railings coming down and it's going in a spiral circle. And then we have these bases that are between them, and those are kind of like the treads on the stairs. So this again is the sugar phosphate backbone, and then the bases are going to be on the interior of this double helix. We saw an alpha helix with the proteins, and the side chains were on the outside. In the double helix, the bases are on the inside. So there is base pairing. There are complementary bases, and this is why there's always the same number of A and T or C and G. Complementary bases are pairs of bases in nucleic acid structure that can hydrogen bond to each other. So if we look at thymine and adenine, we see that one's a purine and one's a pyrimidine. This one's got the single ring and that one has the double ring. And we'll notice that there's always a purine-pyrimidine pairing. You're never going to have two pyrimidines or two purines, because this guy is bigger, isn't he? A and G are about the same size, and C and T are about the same size. But if we pair C with T, the combination is going to be significantly smaller than if we paired A and G, the two larger ones. And in order for that double helix to form, there are pairs of bases, one on each strand, and they're hydrogen bonding, that's what's holding it together, and they need to be roughly the same size so that it can be uniform. It's hard to make a staircase when some of the treads are much longer than the others. They all need to be the same. So the complementary bases come down to size and hydrogen bonding. So with the size, we're always going to have a purine-pyrimidine pairing. And then, is it thymine with adenine or is it thymine with guanine? Well, it's going to be thymine with adenine because these two can form two hydrogen bonds. If thymine pairs with guanine, it can only form one hydrogen bond. And over here, the cytosine and guanine, when those pair up, they can form three hydrogen bonds. Whereas if cytosine forms with adenine, it's only one hydrogen bond. So we get much better hydrogen bonding when we have C and G paired and A and T paired. And that's always, always how it is. And we need to remember that. Your book has a couple of different little methods. So here's C and G, A and T. Let's just look at the letters. Both C and G are curvy letters, right? They look a lot alike. They go together. A and T, I didn't draw that A very well. A and T are angular. They only have straight lines. The A and the T go together. That's one way to remember it. At also spells a word. You can't make a word with A and any of the other letters. Ga is not really a word. So that's one way to remember. But we need to remember A and T, C and G. So the DNA has complementary strands. Those strands are going to match up so that the base on one strand pairs with a complementary base on the other strand. And the whole thing is being held together by hydrogen bonds. What this means is the two strands in DNA are not identical. They're complementary. But they're going head to tail, matching up complementary strands. They are definitely not identical. They're not even just mirror images of each other. If we know the sequence of one strand, we can predict the sequence of the other strand because we know what it will pair up with. By convention, we always list them from 5' to 3' end. But when you're generating a complementary sequence, it is okay to reverse the directionality as long as you specify it. So let's look at this example. Predict the sequence of bases in the DNA strand complementary to the single strand shown. So generally when we're doing something like this, we're going to write the second one backwards. But we need to indicate, okay, this is the 3' end and this is the 5' end, because that's how they're going to be matched up. So what base pairs with A? T. So there's going to be a T first at the 3' end, and then what's next? Another T. Because the A and the T are going to hydrogen bond well together. And A and T, and then here we've got T, so on the other one we must have A. And then with G we'll get C, and with C we get G. C, G, A. So a little bit of things to remember there, but not too bad, right? So got to remember that. So if you're given one sequence, you can write the sequence of the other. Now if you wanted to take this strand and write it in the more normal form, doing 5' end to 3' end, you would just mentally turn it around and reverse the sequence, right? So it would be A, G, C, T, G, C, A, T, T, and this is the 3' end. But this strand is definitely not the same as that strand. Any questions? This is something you definitely need to know how to do.